皆様、こんにちは。本日はバイオセトジェのウェビナーにご参加いただき、誠にありがとうございます。本日、司会を務めさせていただきます、バイオセトジェの長州と申します。どうぞよろしくお願いいたします。本日のウェビナーのテーマは、アルツハイマー病治療開発。将来のアプローチから TFR1 を消した二重特異性抗体争奪による血液脳関門透過の新たなパラダイムへです。それでは本日の講演者をご紹介いたします。本日の講演者は、Biocytogen の贺春辉博士です。贺博士は、同性大学にて博士号を取得され、神経系の発生に関わる細胞、分子メカニズム、および神経、新生疾患の病態メカニズムを専門として研究されてきました。これまで NSFC や国家重点研究開発計画をはじめとする複数の大型研究プロジェクトに参加し、その研究成果は Science Advances, Cell Death and Disease, Cerebral Cortex などの国際的な学術誌にヒット注射または共同ヒット注射として発表されておきました。また、神経変性疾患や疼痛をはじめとする中枢神経系疾患、さらに自己免疫疾患を対象とした前臨床モデルの構築、およびインビボ薬理試験についても豊富な経験を有しています。2025年3月にバイオセトジェンを入社後は、弊社の神経薬理プラットフォームを構築担当して、プロジェクトの実施管理に加え、チーム立ち上げ及び人材育成にも関わっています。本日の講演では、アルツハイマー病治療薬開発における主要な課題、TFR1 に消した脳内争奪技術の最新の動向、バイオセトジェンが開発したヒト化 TFR1 アルツハイマー病モデル、そして TFR1 を消した二重特異性抗体の脳内争奪に関わる前臨床評価データについてご紹介いたします。さらに、バイオセトジェンの神経薬理プラットフォームを活用することで、アルツハイマー病をはじめとする CNS 創薬をどのように加速できるかについてもお話しいただきます。それでは、贺博士にご講演いただきます。なお、ご講演中のご質問とかございましたら、Q&A 欄よりお気軽にお寄せください。また、ウェビナー終了後にもメールにてお問い合わせいただけます。いただいたのご質問がついては、弊社の担当者より改めて回答させていただきます。それでは、皆様、最後までどうぞお楽しみにしてください。贺博士、よろしくお願いいたします。
My name is Chunhui He from Biocytogen, and I'm very pleased to have this opportunity to introduce our clinical platform for neurological disease research. Today, I will mainly focus on our Alzheimer's disease platform. I hope this presentation will give you a clear overview of how Biocytogen can help accelerate your neuroscience drug discovery and progress. I will briefly introduce the key changes in Alzheimer's disease drug development and our TFR1-mediated pre-delivery strategy, and humanized TFR1 AD models, and our integrated neuropharmacology platform for preclinical validation of novel therapeutics. Before introducing our platform, let me briefly introduce Alzheimer's disease. Alzheimer's disease is a complex and multifactorial neurological disorder involving genetic, metabolic, and environmental factors. The major pathology hallmarks include extracellular amyloid beta plaques and intracellular neurofibrillary tangles composed of hyperphosphorylated tau protein. In addition to Aβ and tau pathology, multiple mechanisms contribute to disease progression, including neuroinflammation, oxidative stress, selective dysfunction, and neural loss. Clinically, AD is characterized by progressive connective decline, especially memory impairment, which eventually leads to loss of daily living ability. Due to the complex pathologies and limited treatment options, developing effective therapies remains a major challenge in Alzheimer's disease. After understanding the complexity of Alzheimer's disease, let's briefly review the current therapeutic approaches. AD treatment have mainly focused on the simple proleptic improvements. For example, the NMDA receptor agosis can provide temporal benefits by improving neurotransmission and cognitive function, but they do not significantly modify disease progression. With a better understanding of AD pathology, therapeutic strategies have shifted toward disease-modifying approaches, including the Aβ production, preventing Aβ aggregation, and promoting Aβ clearance through active or passive immunotherapy. Recently, Aβ monoclonal antibodies such as nontalilab and donanemab have demonstrated clinical benefits by reducing amyloid plaque burden and slowing cognitive decline. With the increasing global burden of Alzheimer's disease, the development of effective therapy has become one of the major focus in the pharmacology industry. As shown on the left, Alzheimer's drug development pipelines are expanding across multiple therapeutic areas, including amyloid, tau, neuroinflammation, selective function, and other disease-related pathways. In recent years, the approval of anti-amyloid antibodies has demonstrated the potential of disease-modifying therapeutics and further accelerated innovations in this field. Meanwhile, the Alzheimer's disease drug market is expected to continue growing significantly in the coming years, reflecting the strong unmet medical needs and commercial opportunities. However, despite these advances, the key challenge remains achieving significant drug approach in the brain while meeting safety and efficacy. Although Alzheimer's disease research has made significant progress, developing effective CNS therapeutics remain highly challenging. There are two fundamental reasons behind this challenge. First, the brain is one of the most complex organs in the human body. It contains billions of neurons and highly intercorrelated networks, and disease progression involves multiple pathology mechanisms, including amyloid pathology, tau dysfunction, neuroinflammation, and neuronal degeneration. Second, the brain is protected by the blood-brain barrier, which is essential for maintaining brain homeostasis, but also significantly limit drug delivery. So the blood-brain barrier allows essential nutrients to enter the brain while restricting the penetration of many therapeutic molecules, including antibodies and other biologics. So as a result, many promising drug candidates show limited efficacy because insufficient amounts of therapeutic can research the target site in the brain. And to support the development of novel Alzheimer's disease therapeutics, Biocytogen has established a comprehensive portfolio of preclinical Alzheimer's disease models. These models cover multiple disease mechanisms, including amyloid pathology, tau pathology, neuroinflammation, and cognitive impairment. For amyloid related studies, we provide several widely used transgenic models such as 5FAD and humanized TF1AD models, which enable evaluation of Aβ pathology, therapeutic efficacy, and pre-delivery strategies. In addition, we also have established models for other disease mechanisms, including STZ-induced AD models, Aβ oligo injection models, and LPS-induced neuroinflammation models. These models can be combined with multiple readouts, including behavioral assessment, pathological analysis, biomarker detection, and inflammatory profile to support comprehensive preclinical evaluation. This slide shows the disease progression of our 5FAD model, which is widely used in the Alzheimer's disease. At Biocytogen, we have established a comprehensive phenotype profile in both female and male mice, covering amyloid pathology, neuroinflammation, glycolysis, and cognitive impairment. Here we show the histopathological progression of our 5FAD model, both Aβ plaque burden and astrocytes activation increase with age, and the pathology is more pronounced in female mice. This slide shows the MSD quantification of soluble and insoluble Aβ14 and Aβ14/2 in the hippocampus and cortex of female 5FAD mice. As the disease progression, all four biomarkers increase significantly in both brain regions, demonstrating robust and progressive amyloid pathology. We also support CSF biomarker analysis. Using the MSD, we codified the Aβ14 and Aβ14/2 and the Aβ14/2 to Aβ14 ratio, providing clinical relevant biomarkers for PK and efficacy evaluation in Alzheimer's disease studies. Here we evaluate list building behavior in 5FAD mice. Compared with the wild-type mice, 5FAD mice showed significantly reduced list building scores, indicating the progressive impairments in daily living behavior and cognitive function. We also evaluate the cognitive function using the Morris-Watermaze test. We can see the female 5FAD mice exhibited significant and progressive deficits in spatial learning and memory from 6 to 10 months of age, providing a reliable behavior endpoint for efficacy studies. This slide shows a representative efficacy study using the 5FAD model. Following treatment with FP-ZM1, mice demonstrated significant improvements in spatial learning and memory in Watermaze test, as well as better list building problems. The histopathology analysis also revealed a reduction in Aβ plaque burden in both hippocampus and cortex. Building on this platform, we have also established efficacy evaluation systems for therapeutic antibodies, including Aβ antibodies and blood-brain shuttle based bispecific antibodies. Here we use the lecanemab as a positive control in the 5FID model. Treatment significantly reduced Aβ pathology and improved behavioral performance, demonstrating the robustness of our efficacy evaluation platform. And so we introduced our 5FID model, which is a widely used transgenic model for evaluating amyloid pathology and therapeutic efficacy in Alzheimer's disease. However, when we evaluate the next generation of biologics, especially antibody-based therapeutics, an important elimination needs to be considered. The blood-brain barrier represents a major challenge for drug delivery to the brain due to this highly selective permeability. Only a very small fraction of pre-clinical administered antibodies can enter the CNS. So therefore, conventionally, AD models such as 5FID are suitable for evaluating small molecules or antibodies with sufficient brain exposure, but they may not actually reflect the brain delivery efficacy of novel brain shuttle antibodies. So to better evaluate this next generation therapeutics, model with humanized BBB transport system are required. So to overcome the limitation of the blood-brain barrier, receptors mediate the transcytosis has emerged as a promising strategy for improving CNS drug delivery. Among the different transport systems, transferrin receptor 1 or TFR1 is one of the most extensively studied targets because it is naturally expressed on brain endothelial cells and plays an important role in area transport across BBB. So under pathology conditions, transferrin binds to TF1 on the broad side of endothelial cells, undergoes endocytosis, and is transported across the endothelial layer before being released into the brain side. And so by utilizing this natural transport pathway, therapeutic molecules can be engineered to bind TF1 and hit high across the BBB, enabling improved brain exposure. So, however, successful TF1-mediated delivery requires careful optimization, including appropriate binding affinity, epitope selection, and pH-dependent release, and ligand density to achieve efficacy in transport while maintaining normal TFR function. So therefore, TF1 has become one of the leading approaches for developing next-generation brain shuttle therapeutics. After understanding the mechanisms of TF1 mediates the transport, let's look at how TF1-based brain shuttle antibody works. In this strategy, a bispecific antibody is designed with two functional domains. One arm binds to TF1 expressed on brain endothelial cells, while the other arm targets the therapeutic targets such as amyloid Aβ. After intravenous administration, the TF1 binding domain enables the antibody to attach to the BBB endothelial cells and enter the transcytosis pathway. The antibody is then transported across the endothelial cells and released into the brain parenchyma. where the therapeutic arm can engage its target and exert its pharmacology effect. So this approach provides a potential solution to overcome the limitation pre-exposure of conventional antibodies and improve the efficacy of CNS biologics. Beyond its psychological role in aerial transport, TfR1 has also emerged as an attractive target for drug delivery across the blood-brain barrier. By utilizing TfR1-mediated transport, the so-called transport vehicle technology enables the delivery of various therapeutic modulators, including antibodies, proteasomes, and oligonucleotides into the brain. Among these approaches, antibody-based brain shuttle technology has attracted significant attention, especially for Alzheimer's disease. For example, trontinemab, developed by the Roche, is an Aβ antibody engineered with a TfR1 binding shuttle molecule based on the technology platform to enhance the BBB penetration. This strategy aims to increase the antibody exposure in the brain while maintaining target engagement and therapeutic activities. These advances highlight the importance of translational models that can actually evaluate TfR1-mediated brain delivery and therapeutic efficacy. Although Aβ antibodies have demonstrated clinical benefits, their therapeutic potential is still limited by the insufficient brain exposure and safety concerns. The traditional Aβ antibodies administered IV have very limited ability to cross the blood-brain barrier. Therefore, achieving sufficient amyloid clearance often requires high systemic exposure, which may increase the risk of adverse events, including ARIA. Brain shuttle technology provides a potential solution by incorporating a TfR binding molecule into the antibody design. By engaging the TfR on brain endothelial cells, the antibody can utilize receptor-mediated tricytosis to enhance brain delivery, allowing improved target engagement with potential lower systemic exposure. Trontinemab represents one of the clinical examples of this strategy, coming Aβ antibody with the TfR-mediated brain shuttle modulates to improve CNS delivery. These advances highlight the importance of developing translational models that can accelerate the evaluation, both brain penetration and therapeutic efficacy of TfR-based therapeutics. So to evaluate the TfR1-mediated brain delivery therapeutics, a humanized TfR1 model is required because TfR interaction is spatially dependent. In the TfR1 mouse model, the extracellular domain of mouse TfR is replaced with a human TfR1 counterpart, while maintaining the physiological expression of pattern. This enables human TfR1 targeting antibodies or brain shuttle molecule to be evaluated in a more clinical relevant mouse model. So therefore, the humanized TfR1 mice provide a valuable platform for assessment being blood-brain barrier transport and CNS drug delivery. The humanized TfR1 mouse model provides several advantages for evaluating TfR1-mediated drug delivery strategies. First, the model uses full genomic lock-in strategy to introduce human TF1 sequences while maintaining physiological expression patterns. Second, the humanized TF1 protein has been confirmed to maintain normal physiological functions, providing reliable translational models. With these advantages, the humanized TF1 mice can be applied for BBB penetration studies, safety evaluation, and in vivo efficacy assessment of TF1 targeting therapeutics. Especially for pre-shuttle antibodies, this model enables a more clinical relevant evaluation of drug delivery and therapeutic potential. Before applying the humanized TF1 model for drug evaluation, we first confirmed its basic physiological characteristics. The humanized TF1 mice showed normal body weight growth patterns comparable to control mice, both males and females, across different ages. These results indicate that the humanized TF1 does not cause obvious abnormalities in general health and or development. So therefore, the humanized TF1 mice provide a reliable and physiologically relevant model for evaluating TF1 targeted therapeutics. Next, we confirmed the expression profile of TF1 in the humanized TF1 mice. Western blot analysis showed that TF1 expression patterns across different tissues were comparable between the humanized TF1 mice and the control mice, indicating that humanization did not disrupt the physiological expression pattern. Importantly, flow cytometry analysis demonstrated that human TF1 was specially detected in brain endothelial cells from humanized TF1 mice, while it was absent in wild-type mice. So these results confirmed the successful expression of human TF1 mice in the relevant blood-brain cell population, providing the suitable platform for evaluation human TF1 targeting brain shuttle therapeutics. In addition to confirming human TF1 expression, we further evaluated whether TFR1 humanization affects its physiological function. Since TF1 plays an important role in area transport and homeostasis, we analyzed multiple serum area-related parameters in human TF1 mice. The results showed that the serial area levels, TRBC and TRBC levels or other indicators were comparable between humanized mice and wild-type mice. So this finding demonstrated that humanization of TF1 does not cause significant abnormalities in area metabolism. So after confirming the suitability of the human TF1 mice model, we further evaluated its application for assessment BBB penetration of human TF1 targeting antibodies. So in this study, the human TF1 mice were administered with TF1 antibodies, and brain uptake was evaluated through multiple approaches. Sample including serum, total brain, and brain regimen after capillary depletion were collected for bioanalysis. In addition, histological analysis such as immunohistochemistry or immunofluorescence can be used to visualize antibody distribution in the brain. These approaches allow us to distinguish true brain penetration from antibody remaining with brain vacuolate. So therefore, the humanized TF1 mice provide a reliable platform to quantitatively evaluate BBB transport efficacy of TF1-mediated brain shuttle therapeutics. Using the humanized TF1 model, we further quantify the antibody distribution in different compared traits of the brain. After administration of anti-TF1 antibodies, the antibody level was measured in serum, whole brain, and brain regimen using the MST-based bioanalysis. Importantly, the detection of antibodies in brain regimen demonstrated that the antibodies were able to cross the blood-brain barrier and enter the brain tissue, rather than only remaining within the brain vasculature. The results confirm that the humanized TF1 mice can effectively differentiate and quantify BBB transport efficacy of human TF1 targeting antibodies. So this demonstrated the value of humanized TF1 mice as a translational model for evaluating brain shuttle therapeutics. After validating BBB penetration in humanized TF1 mice, we further evaluating brain delivery of a brain shuttle antibody in Alzheimer's disease model by combining the humanized model with the 5FAD model. We establish a double humanized AD model that enable evaluation of both human TF1-mediated brain delivery and amyloid pathology. Following administration of trontinemab, the human IgG signals were detected in the brain, demonstrating successful BBB penetration. Importantly, the antibody signal showed a co-localization with Aβ plaques, indicating that the delivered antibody was able to reach its pathology target within the brain. So these results demonstrated that the TG5FAD and the human mice model provides a translational platform for evaluating brain shuttle therapeutics from BBB penetration to target engagement. And to support the development of BBB penetrating therapeutics, we established the TG5FAD and the human TFR mice model. Importantly, humanizing TFR1 does not alter the Alzheimer's disease phenotype. Similar to the 5FAD model, these mice inhibit progressive Aβ pathology, elevated Aβ14 and Aβ14/2 levels, and robust cognitive deficits. This provides a reliable preclinical platform for evaluating both brain penetration and therapeutic efficacy of TF1 targeting antibodies. This slide demonstrates the application of our TG5FAD and human TF1 model for evaluating brain shuttle antibodies. Following treatment with trontinemab analog, mice showed a significant improvement in the nest building test performance and spatial learning and memory compared with the vehicle group. In addition to behavioral improvement, trontinemab analog also produced significant pathology benefits. Treatment reduced Aβ deposition in both the cortex and the hippocampus, as confirmed by immunohistochemistry, and also improved brain and CSF Aβ biomarkers. So together, these findings demonstrated the therapeutic efficacy of the brain shuttle antibody in our TG5FA and human TFR1 mice model. So beyond the humanized AD models, the Biocytogen also provides integrated preclinical service to support the Alzheimer's disease drug delivery from targeted validation to therapeutic evaluation. Our capabilities cover the entire workflow, including disease modeling, different administration routes, behavior assessment, sample collection, and downstream analysis. For AD studies, we can perform various in vivo models with different delivery approaches, including systemic administration and intracerebral injection models. We provide comprehensive efficacy evaluation through behavior tests such as Morris water maze, Y maze, and novel object recognition, combined with pathological, biochemical, and molecule analysis. With our integrated platform, we can support comprehensive preclinical evaluation of innovative Alzheimer's disease therapeutics. And our platform include a wide range of preclinical models. We focus on three major areas: neurodegenerative disease, pain, and Parkinson's disease. In addition, we also work on the neuromuscular disease, autism, and other neurological disorders. In addition to our disease models, we offers a comprehensive behavior testing platform. We support a wide range of behavior assessment covering motor function, sensory function, coordination, pay, gait, and neurological deficits. This slide highlights our behavioral assessment for emotion and cognition. We provide a comprehensive panel of tests evaluating learning and memory, anxiety and depression-like behaviors, social interaction, and activity of daily living. This assay can be combined to support efficacy evaluation across a wide range of CNS disease models. And our experienced in vivo team support multiple drug administration routes from conventional systemic dosing to CNS-targeted delivery, enable flexible and customized study designs for a wide range of clinical programs. And our platform support precise brain region dissection and high-quality CSF collection in both mice and rats. These capabilities provide reliable samples for biomarker analysis, PK and PD evaluation, and target engagement studies. This slide provides an overview of our histopathology capabilities for CNS studies. We offer a comprehensive pathology workflow, including routine histology, HCIF, and TSA-based immunofluorescence. Beyond high-quality staining, we also provide a quantitative image analysis using the Halo software, enabling objective assessment of biomarker expression, cell coating, fluorescence intensity, lesion and lesion area, and co-localization analysis. We also offer the comprehensive cytokine and biomarker analysis using MSD, Luminex, ELISA, and CBA. Our platform supports a wide range of CNS biomarker in serum, plasma, and tissue samples from both mouse and human. This example demonstrates our MRI-based ARIA assessment platform. We can reliably detect both ARIE and ARIH in AD mouse model, providing valuable support for the preclinical safety evaluation of Aβ antibodies. That's all. Thank you.
お忙しいところ、バイオセツルのウェビナーにご参加いただき、誠にありがとうございました。以上をもちまして、ヘ博士によるプレゼンテーションを終了させていただきます。ご不明点やご質問などがございましたら、どうぞお気軽に info@biocytogen.com.cn までご連絡ください。また、QR コードをスキャンいただけますと、弊社の公式ウェブサイトをアクセスいただけます。弊社のサービスや製品に関するより詳細な情報をご覧いただけます。今後ともBiocytogenをどうぞよろしくお願いいたします。 次回のウェビナーで皆様とお目にかかれることを心より楽しみにしております。どうもありがとうございました。